As we said earlier in this lecture, yeast can be engineered to produce certain substance such as insulin. However, it can also be engineered using recombinant DNA technology to express artificial pathways for the production of fuels, biopharmaceuticals and perfumes. These so-called cell factories can cost up to $50 million US. We will take a look at a few of these examples later in the lecture. Yeast and other unicellular eukaryotes often have poor gene expression or complex regulation. Therefore, they must be engineered to contain some optimal properties, which can be useful in industry and thus referred to as their own gene factory. Yeast is a so-called platform organism and can be used to generate a range of different products. Most of these techniques often struggle to make the transition between lab and industry. This gap is referred to as the valley of death. The main aim of metabolic engineering and synthetic biology of yeast is optimal performance of yeast strains. One of the main unicellular eukaryotes used in this process is yeast. Yeast is currently being manipulated by a scientist called Jens Nielsen to grow at a higher temperature. As you probably know, yeast currently struggles to grow at a temperature above 35 degrees. This heat shock response is as a result of evolution. Therefore, it may seem obvious to some of you that in order to grow yeast at a higher temperature, we must mimic evolution artificially. This is called high temperature adaptation. If we could manufacture yeast to grow at higher temperature, it would be much more effective since the cost of cooling would be decreased and the bioreactor would be much more stable, as you could also combine the yeast clones with diverse enzymes that prefer higher temperatures. Therefore, in recent studies directed at adapted lab evolution aimed to grow yeast at 39.5 degrees, which is significantly higher than optimal 35 degrees. The yeast was grown artificially at gradually higher temperatures for around 600 generations to eventually produce clones of the yeast that would grow better at 39.5 degrees. These successful yeast strains produce almost twice the amount of glucose at this elevated temperature. This therefore also increased the production of our industrial products. Asking yourself, how did this genetic mutation occur? The answer is somewhat complicated, however very interesting from the point of recombinant DNA. Upon analysis of the yeast clone, a duplication was discovered on chromosome 3. There was an accumulation of point mutations on the E or G3 and ATP3 regions. However, when E or G3 was removed and only an ATP3 mutation present, no change in yeast was observed. On analysis of the E or G3 mutations observed on all of the clones, there's a dramatic increase in growth, as you can see from the graph. This causes a shift in the sterol concentration, phycosterol, which is a bended sterol accumulated in the yeast. So it was concluded that changing the sterol composition brings about the ability for yeast to grow at different temperatures. This adaptability has a huge translational impact on the production of yeast, as the wild type produces less ethanol than the mutated strain. This experiment supports our discussion on the importance of recombinant DNA technology.